GS signaling pathway. GPCR, or G-protein-coupled receptor, is a large family of cell surface receptors that play a crucial role in cellular signal transduction. GPCRs span the cell membrane 7 times, giving them the name 7-transmembrane receptors. The extracellular part of the GPCR binds to signaling molecules, ligands. The part inside the cell interacts with G proteins to transmit the signal. A ligand, which may be a hormone or neurotransmitter, binds to GPCR, the receptor becomes activated. The activated receptor promotes the exchange of GDP for GTP on the alpha subunit. This leads to the activation of alpha subunit. Activated alpha subunit dissociates from beta-gamma dimer and moves towards adenylyl cyclase, which is embedded in the membrane. Alpha subunit activates adenylyl cyclase, which converts the ATP into CMP. As the adenylyl cyclase is activated, the concentration of CMP increases in the cell. CMP then binds to the regulatory subunits of protein kinase, thus the protein kinase A becomes activated. The activated catalytic subunits then moves towards the target proteins. It can phosphorylate then target proteins, leading to cellular responses such as gene transcription or metabolic changes. The activated pKa then translocate into the nucleus. CRB, CMP response element binding protein, is a transcription factor that plays a critical role in gene expression. pKa phosphorylates CRB at a specific serine residue, SER133. This phosphorylation is crucial for its activation. The activated CRB binds to specific DNA sequences called CMP response elements, CREs, in the promoters of target genes. CRB recruits coactivators like CRB binding protein, CBP, or P300, which help form a transcriptional complex that initiates the transcription of target genes. Desensitization of GPCRs, G-protein-coupled receptors, refers to the process by which a receptor becomes less responsive to its ligand, signaling molecule, after prolonged or repeated activation. This mechanism helps prevent overstimulation and allows cells to adapt to sustained signaling, maintaining cellular homeostasis. The desensitization of GPCRs is a key regulatory process in signal transduction and involves a series of steps that reduce the receptor's ability to activate downstream signal. Specific kinases, primarily GRKs, G-protein-coupled receptor kinases, phosphorylate the intracellular regions of the receptor, typically on serine and threonine residues. This phosphorylation occurs mainly on the C-terminal tail and intracellular loops of the receptor. The phosphorylation of the GPCR serves as a binding site for arrestins, proteins that inhibit further G-protein activation by sterically blocking the receptor's ability to interact with the G-protein. Arrestins also facilitate the internalization of the receptor, removing it from the cell surface and effectively shutting down the receptor's activity. This reduces the cellular response to the stimulus. Receptor either internalize, endocytosis, or degraded. CMP phosphodiesterase, PDE, hydrolyzes CMP into 5'-AMP, reducing CMP levels. Protein phosphatases remove phosphate groups from proteins that were phosphorylated by pKa. This deactivates the target proteins, effectively shutting down the signaling cascade. As the alpha subunit of GS protein has intrinsic GTPase activity, so it hydrolyzes the bound GTP into GDP. This returns the alpha subunit of GS protein to an inactive state, allowing it to reassociate with beta-gamma dimer and the receptor, so the pathway is terminated. In contrast to the GS signaling pathway, in which stimulatory G protein is involved, the GI signaling pathway involves inhibitory G protein. The alpha subunit of GI protein is activated as by similar procedure occur in GS signaling. Activated alpha subunit dissociates from beta-gamma dimer and moves towards adenylyl cyclase that is embedded in the membrane. Alpha subunit of GI protein cause inactivation of adenylyl cyclase, so ADP is not converted in CMP. Hence the further activation of pathway does not occur and the pathway terminated. Please like, share and subscribe and hit the bell icon for more videos.